All right, so we're going to finish up our talk on the digestive system. So the stomach here is a pouch-like organ, 25 to 30 centimeters long. It's just below or inferior to the diaphragm, more towards the upper left portion of the abdominal cavity. It receives the food from the esophagus, then mixes that food during segmentation, and it mixes it along with gastric juice that helps to break it down. It initiates protein digestion. That is important. Now, remember, you need to know the function. So the function is going to be primarily, well, it's actually going to be a mix of chemical and mechanical digestion here. All right, so it's mixing and churning. All right, but it is also really doing some chemical digestion here. So mixing food with gastric juice. Protein digestion. Protein digestion begins in the stomach. Very little absorption, but it does have some absorption. And then it moves the food on down to the small intestines. The stomach has what we call rugae, which are gastric folds, and these are tissues that actually allow for the stomach to be distended or to stretch. All right. We also have that like in our bladder, but in our stomach it allows our stomach to stretch and allow for that extra room. The stomach has two layers of smooth muscle. Remember, smooth muscle is autonomic or under automatic, so to speak, control. It has an inner circular layer here, and you can see that up here at the top, these inner circular fibers, and then an outer longitudinal and even some oblique fibers as well. So longitudinal, circular, and then here are some oblique, which is kind of a diagonal. And that's going to be more of an inner layer there. Those muscles are really important because they aid in the mixing and churning of the food. There, you've got all those orientations. You've got circular, longitudinal, and oblique, and so they're able to really thoroughly mix everything. The stomach is divided into the cardia, which is here. All right, the cardia here. Then we have the fundus. And now if you have had a child, you might be familiar with the term fundus. Fundus is the portion going above an opening. So when you have a child and you're pregnant, you go in and they measure you to the height of the fundus of your belly, which is going to be the portion above a certain part. That helps them to estimate the size of the baby. So fundus is just a general term. So that is the portion above the opening there, which is where the cardia is. And then we know the body is the main portion here. So the body is the main portion of something. And then we have the pylorus, all right? And so the pylorus is this section here, which is, composed of the pyloric canal and the pyloric antrum. Next, we have the pyloric sphincter. What do we need to remember about that? What did I tell you? You had 3 sphincters. This is number 2, location pyloric sphincter, which is a circular muscle which controls movement one direction, all right? One direction. So that controls gastric emptying, so that is important. Some other things to point out here, you can see the gastric folds here, the rugae, and so be careful when you are looking at this to know, is it pointing to the folds or does it mean the body? So be careful with that with your labeling. We also have the greater curvature and the lesser curvature, all right? All right, gastric secretions. These are some of those important molecules and enzymes that help to break things down. We start with pepsinogen. Gen means beginning. So when you see ogen, that means it's a precursor molecule that is going to be changed into an active form. So we have one form that's inactive, and then something will be added or removed, and then it will be made active by doing so. Okay, so we have pepsinogen, which is secreted by chief cells, and that is the inactive form of pepsin. That becomes activated by hydrochloric acid, and so once it becomes activated, it becomes pepsin. Pepsin begins digestion of proteins. Very, very important. So pepsin is protein. Right here, protein, P and P, pepsin, proteins. You need to know that, all right? So pepsin helps to break down proteins, begins the chemical digestion of proteins. Then we have gastric lipase, ASE, that tells us it's an enzyme, and that is also secreted from chief cells, and that splits fat molecules. So that begins the digestion of fat. This can be weak if you have a low pH. Okay. Next, we have parietal cells of the gastric glands that are secreting hydrochloric acid. Okay, and again, we need that in order for pepsinogen to be activated into pepsin. Mucus, or what is sometimes called mucin, and that's a glycoprotein component of mucus, is secreted from goblet cells. Remember goblet cells from histology? I bet you thought you were going to be able to forget histology from Anatomy 1. Nope, sorry, still got to know that information. Goblet cells secrete mucus, and so this protects and coats the wall of the stomach, and basically keeps it from digesting itself. You've got all that hydrochloric acid, right? And so you don't want that to dissolve the lining of your stomach. So that mucus is very important. We then have intrinsic factor. Intrinsic factor is important, especially for the elderly population. So if you work in a nursing home, this is important information, or if you work in geriatrics period. Intrinsic factor is required for absorption of vitamin B12 from the small intestines. As we get older and our body begins to break down, we don't produce that intrinsic factor. And so taking vitamin B12 supplements orally is not effective. And if we don't have our vitamin B12, that can affect our red blood cells and many other things. All right, so vitamin B12 is important. So for somebody that is in the geriatric component of life, they would need to actually have B12 shots as opposed to oral B12 tablets. So that's an important clinical application there. Regulating gastric secretions is important. We don't want to continuously be making hydrochloric acid or having it be continuously released, because it's going to build up, cause heartburn, cause peptic ulcers, and other problems. All right, so that's just one example. So gastric juice is produced, and it is affected by hormones. All right, we've got sympathetic impulses that are going to decrease gastric activity. So we talked about this earlier, but let's go ahead and highlight that. Sympathetic is going to decrease and parasympathetic is going to increase. One way that occurs is acetylcholine is going to be, and we abbreviate that ACH for acetylcholine, is going to be released by parasympathetic fibers, and it's going to inhibit somatostatin, which then in turn inhibits the acid secretion because it triggers release of pepsinogen. And remember, ogen is that precursor molecule, and so that signals hydrochloric acid to be decreased in the amount that is being secreted. All right. Next, we have gastrin. Gastrin is a peptide or protein hormone that increases the activity of gastric glands, so it thereby increases gastric juice secretion. Parasympathetic impulses can also cause the release of histamine from mucosal cells, and that will stimulate more gastric secretion as well. Cholecytokinin, which is abbreviated CCK, is going to decrease gastric motility. Okay, that's released by the small intestine when the proteins and fat enter the small intestine. And so these are ways that your body knows for your stomach to start working, or your food has moved on, so the stomach stops working, that type of thing. Gastric secretion occurs in 3 phases here. We have the cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase is before food actually reaches your stomach. So this is even before you eat. This is when you smell and see or think about food, and that triggers parasympathetic reflexes. So your mouth starts watering, you start salivating. Okay, gastric food is also secreted in response. So the greater the hunger, the greater the secretion will be. All right. The gastric phase plays the largest role in secretion and begins when your food actually hits the stomach. So when that bolus of food hits the stomach. So the presence of food stimulates the release of gastrin. Okay, so it's going to stimulate the release of gastrin here. Lost my train of thought, which is going to cause more gastric juice to be released, and the pH is going to increase, which triggers more release of gastrin, so then the pH will eventually drop. Okay, if pH reaches 3.9, gastrin release is inhibited. At 1.5, it stops, and so your body has a regulation to know when to stop secreting that. Next, we have the intestinal phase, and this is when food enters the small intestines. So this is when food enters the small intestines. Remember, this one is just thinking about food. So, intestinal phase, when the food enters the small intestine. Food starts to touch the intestinal wall, and that causes the gastrin again to be released, intestinal gastrin here, and inhibits gastric juice secretion. And then protein and fat, when it enters, it is going to cause cholecytokinin, CCK, to be released as well, which decreases gastric motility. So CCK here, let's pick a different color. CCK, which decreases gastric motility, that triggers things to slow down. This is part of the reason why protein and fat satiate you more, okay, because they take longer to digest, and so you feel full longer and you don't feel hungry as quickly. All right, so gastric absorption. So we looked at the majority of digestion occurring in the stomach. Well, the majority, not all, the majority of absorption occurs in the small intestine. Gastric enzymes begin breaking those proteins down in the stomach, but proteins are large and they take multiple steps to break down. And the stomach's just not well adapted to be able to absorb those products, okay? It's not designed for absorption. It does, however, absorb a few things like some water, salt, alcohol, lipid-soluble drugs, all right? So some absorption, just not much. So here we're looking at the actual mixing of the food in the stomach with the gastric juice. And this is where our bolus of food is going to become chyme. Okay, once it becomes liquefied or paste-like, it's going to be called chyme. So the stomach is going to fill, the wall is going to stretch or distend, and then this pyloric sphincter will close. All right, and so that's where we're at here in A. The pyloric sphincter closes so that you have some time to mix the food. Mixing combines food and gastric juice in order to make the chyme, and then peristalsis occurs to move the chyme to the pyloric sphincter, which will then relax and allow some chyme into the duodenum if you prefer. We have a reflex that slows down stomach emptying called the enterogastric reflex. We can also have, chyme goes in the duodenum, causing the intestinal wall to stretch, triggering that enterogastric reflex. It begins in the small intestines. This is where we get the word entero from, and it ends in the stomach, so we get that. It's called gastro. Bacteria like H. pylori can actually enter the stomach and cause peptic ulcers. That's actually a fairly new finding. They used to think ulcers were always caused by stress or by eating, and I don't know, the past 15-20 years we discovered Helicobacter pylori, which is a bacteria, and so you can actually treat that with antibiotics, which is pretty cool. And so the enterogastric reflex can limit parasympathetic impulses reaching the stomach, and so that can decrease peristalsis, and then in turn intestinal filling is going to slow. If chyme has a very high fat content, cholecytokinin, CCK, is released, which further inhibits peristalsis and allows that to be broken down partially. The pancreas is one of our 4 accessory organs. It has cells that release pancreatic juice, and these are pancreatic acinar cells. It has a dual function. It can act as both an endocrine gland and an exocrine gland. So the exocrine function is to secrete that digestive juice, and we call that pancreatic juice. In pancreatic juice, we have enzymes that help us to digest carbohydrates, fats, proteins, and even nucleic acids, all right? These are really important. You need to be sure that you know these, okay? Pancreatic amylase, ASE, tells us that it's an enzyme. This is going to split or break starch and glycogen down into a disaccharide. So it's going to break it down. Then we have pancreatic lipase, which breaks triglycerides, lipids are fats, right? So it breaks triglycerides down into fatty acids and monoglycerides. Next, we have trypsin, chymotrypsin, and carboxypeptidase, and these work together to break down proteins. This is no single enzyme can break down all of the possible amino acid combinations alone. So we've got to have several of these guys to completely digest proteins, and these act in a cascade of events. Trypsin is the active form of trypsinogen, which is the inactive form. That is activated by enterokinase. Trypsin will then go on and activate chymotrypsin, which will digest proteins, and then that will go on and activate carboxypeptidase. And that's what it says under my little picture here. It's going to activate carboxypeptidase, peptide proteins, okay? And so trypsin, chymotrypsin, and carboxypeptidase all work together, all right? Nucleases, well, that one's pretty easy. That breaks down nucleic acid. That's one of the handy things about enzymes. Their names often tell you what they do, right? Okay, so then lastly here we have bicarbonate ions. This is an important component of the bicarbonate acid buffer system in our body. Here, these bicarbonate ions are going to make the juice more alkaline to allow those digestive enzymes to function and not be denatured by our stomach acid. So we've got to, of course, regulate pancreatic secretion as well. So hormone secretion the hormone secretin is going to stimulate the release of that pancreatic juice when chyme enters the duodenum, all right? And this is high in those bicarbonate ions. Pancreatic digestive secretions are going to be regulated by cholecystokinin whenever those proteins and fats enter. And that stimulates the pancreas to release pancreatic juice that is high in enzymes. So the liver is another accessory organ we have here. This is actually the largest internal organ of the alimentary system. It is located in the upper right quadrant just beneath the diaphragm, and it is broken down into lobes. When you are looking at this, especially in lab, you want to pay attention to the orientation. Specifically, look at the location of the gallbladder. So you know if you're looking inside, from what side you're looking at, okay? I wanted to say superior and inferior, but that's really not correct. All right, so here, look down here. Here's the gallbladder. So when the gallbladder is beneath, then that is the right lobe. Okay, that is the right lobe. Get my pen to work. The right lobe. And when the gallbladder is fully showing, then you can see here at the top, we still have the right lobe here, but it's going to be really important when you look at the quadrate lobe and the caudate lobe. Right, so really important to look at that because the right and left lobe are fairly easy to differentiate because of size, but the quadrate and caudate can be difficult to differentiate from each other. The liver has a ton of functions, so many functions. Some of these include maintaining blood pressure concentration, glucose concentration, carbohydrate metabolism, and this is done by responding to insulin and glucogen and lowering blood glucose by breaking down glycogen and vice versa. Formation of urea, secretion of bile. It oxidizes fatty acids, deaminating proteins, which basically just means taking a nitrogen off. So that's an amine group has nitrogen. It stores vitamins like A, D, and B12. It participates in iron homeostasis, so regulating iron. Detoxification is a biggie for the liver. It removes things like alcohol. It also acts as a blood reservoir, which is really important. So it does so many things.